Hi there, this is a quick help video for the Enzyme Lab. And in the Enzyme Lab, when you open it up, you'll see there's an upside down graduated cylinder. There's the reaction chamber, which contains hydrogen peroxide and the enzyme. And then you have different parameters you can set over here on the side. So the main thing is here, not so much to worry about any specific measurement at any specific time, but just to sort of observe the overall trend. So kind of quickly, you would just select the parameters. So there might be concentration, for example. I'm going to just pick a concentration here. You should kind of look at all of them, and then you can compare it to the graph when you're done. And you're going to start. So what I'm looking at here is a specific concentration of the enzyme. So this is 100 microliters. I'm going to go ahead and start. So I'll talk a little bit. And what happens is when hydrogen peroxide comes in contact with the catalase enzyme converts it to water, a little bit of heat, and oxygen gas. When the oxygen gas is created, it can be captured in this nozzle here. So it actually comes out of the chamber. So this sort of tubing here is to direct it up into the graduated cylinder. And as it bubbles, it displaces the water that's in there. So we're actually measuring the oxygen gas that's produced. So we can kind of do different things, speed it up a little bit if you like. You can pause it if you would like. You can rerun it over here. So let's speed it up quite a bit. I'm going to go ahead and just stop it. Normally we run this about 10 minutes in the lab. So we just gave you that option to kind of speed it up. You don't get to do that in real life. So online you get that special sort of speeding up the time. And then when you get finished, you can view the graph, and the graph is going to compare these different concentrations that are here. I'm going to go ahead and click on that. And let me see if I can scroll up here just a tiny bit. There we go. So that's going to compare those different concentrations. And so again, it's not critical that you remember a specific volume, but just get an overall viewpoint. So when we interpret the graph, this is the most important part of this lab. So we have the simulation there, but in terms of the final output, the graph is really, really critical. A couple things we want to look at. So the graph, as you see in the higher concentration, really takes off. The lower one, because there isn't as much enzyme available, runs a little bit slower there. And you can see the two low ones, that 50 and 100 microliters, they're pretty steady and slow over time. As you increase concentration to a point, generally you tend to speed up the concentration of the reaction. Now that has its limits because you only have a certain amount of hydrogen peroxide in the chamber. And so that really takes off. But over time, you start to see them both flatten out and about the same rate at about 10 minutes. And again, there's a limit to how much hydrogen peroxide you can process because there's only so much in the chamber. So when you see this curve start to flatten out, keep in mind the enzyme is being recycled over and over and over again. And the enzyme here is catalase. So that is recycled, recycled, recycled. So the reason this flattens out actually doesn't have anything to do with the enzyme availability as much as the hydrogen peroxide starts to run out there. And so as you go through the other sort of setups here, and again, you can read the lab to kind of read a little bit about the background. You could try different temperatures and, run those as well. And again, you can go, speed up the time if you would like, and pH, and compare. So let's look at one where you really see a huge impact. With concentration, we always get some sort of reaction. But with, as you get to kind of temperature and pH, we get to what's called extreme conditions. And you can really see those start to slow down a lot. Again, let me move that up a little bit. There we go. So you can see at the lower ones, you don't get as much activity. And let's compare that to pH. And we'll pick something like pH 2. Oops, I didn't want to go quite to view graph yet. We're going to start timer. And with this one, you don't see a huge displacement. So I know it looks like there's a lot of bubbles here being made because it's being sped up, but actually if we slow it down, it's a quite slow reaction. So don't worry about the number of bubbles so much as looking at that displacement. So now let's go and look at the graph. And again, like the other graphs, it's showing all the different, whether it's concentrations or enzymes. Here is pH. So here you see again that kind of flattening that we saw before at pH 7. pH 7 is a neutral pH, so that's fairly close to mimicking what we would see in the body. And catalase is an enzyme found in different organs in the body and blood. However, when you get to the low pH, you see there's not really much activity at all. And so we want to now think about that in terms of the idea of denaturing. So what happens, and this is a very acidic pH, what happens in very extreme conditions to enzymes? So you could do a little bit of reading in the lab itself so that you can kind of think about what's happening there. That's about it. So just trying to give you a little bit of an idea of how this relates to real life and what we do in the lab. So again, that's the chamber where you have the enzyme combined with what we call the substrate, and here that's hydrogen peroxide. And then we have a way to sort of capture the bubbles. So our product here is water, which we can't really see. It just looks like the same clear liquid that hydrogen peroxide does, and that stays in the chamber. However, one of the products is oxygen gas, and that we can measure because we can measure the bubbling that goes on there. And so the bubbling will displace a certain volume of water that's already in the inverted graduated cylinder, and we can get a sense of how fast the reaction is occurring. Okay, that's about it. Please read the manual and contact your instructor for any other questions. Have fun.